ISCB SC RSG-Türkiye, and we are a society under the International Society for Computational Biology. This society is based on the USA, and this society has a section called ISCB Student Council, which consists of many regional student groups. And RSG-Türkiye is one of these regional student groups. And our student group was founded in 2011. And this year we celebrate our 10th anniversary. And this student group was founded in 2011. And since then, we had a bunch of leaders since 2011. And as RSG-Türkiye, we organize many events. Our main event is our annual symposiums. And we were organizing our symposiums face-to-face until 2019. And this picture is the last our face-to-face workshop. And after that, we had to organize our symposiums online. And we also organize webinars. Until now, we have organized more than 50 webinars. We have had more than 1,000 attendees from 40 countries, and we have more than 300 members on BioInfonet channel. And we also translate our webinars in our YouTube channel from English to Turkish. And some of the webinars were already translated into Turkish. And in our website, we have a section called Resources. We collect resources related to the computational biology into this website. We try to keep this website updated and open source, and anyone can suggest anything to this website. And in our website, we have a blog section which our other members or other people can share their prepare some content about computational biology, like some short reviews or some short tutorials, etc. And you can also, if you want to share some content like this, you can ask us and we can allow you to share this kind of content in our blog section. And we also organize a bunch of workshops. This was one of our workshops, NCAPI, in 2019. We aim to teach Python to other people. And also with our annual symposiums, we organize we try to add some workshop sessions. For instance, this session is from our last year's workshop. We had workshops like this one, Introduction to R, workshop on machine learning with R, and data visualization by using ggplot. So we are organizing a bunch of workshops, and we are also organizing workshops other than the symposiums like this one. And before this one, we have also organized this workshop for single cell analysis to introduce single cell RNA-seq and single cell ATAC-seq. And we have also organized some panels as a live stream on YouTube, like to discuss about AlphaFold, or to discuss with some high school students who have worked on SARS-CoV-2 virus by using some machine learning methods. And in YouTube, we also organized career fairs to discuss about previous and current experiences of our guests. And we have also organized some Instagram live sessions to discuss where and what do bioinformaticians do. And as RSG-Türkiye, we organize journal clubs. It takes place every month, and we keep organizing our journal clubs. And we believe we are excellent community to be a part of. If you want to be a part of RSG-Türkiye, we are always open to everyone. You can register by using our main website link. We are active in Twitter, Instagram, Facebook, YouTube, and LinkedIn. By using this link and this QR code, you can access to our social media accounts and our main website link. From our website link, you can find the registration link, and we will allow you to join RSG-Türkiye. And in RSG-Türkiye, we use Slack as our main communication tool. And we also allow you to join our Slack workspace after your registration. And as an upcoming event, we have a last journal club next Saturday. And we are also planning to organize a bunch of journal clubs in 2022. And this is our schedule for this weekend's workshop. After my presentation, Dr. Burak Veli Kabasakal will talk about the experimental techniques. And then Cemil Can Saylan will show you the important websites about structural biology and visualization with PyMol. After lunchtime, Dr. Salia Ajuner will talk about molecular docking principles. And then Eda Şamiloğlu and Feyza Maden will show you some structural biology tools like HADOC, PRISM, SwissDoc, and AutoDoc. Without further ado, I would like to introduce you Burak Veli Kabasakal. He's an assistant professor at Turkish Accelerator and Radiation Laboratory, Ankara University, and he will talk about experimental methods for identification of protein structure. Dr. Burak Kabasakal, stage is yours.
Thank you very much. Good morning, everyone. Actually, I'm impressed what you have done already, and happy anniversary. I didn't know that. And also thank you for organizing this event and inviting me. Let me start share my screen. Okay, you can see my screen, I think. Okay.
Yes, we can see.
And also thanks to the participants for being here on an early Saturday morning. Okay, let's start with discovering experimental techniques. Actually, there are many things to discover, but I will mention some of them. And before starting discovering the techniques, let's talk about protein synthesis, how the protein synthesis occurs in the cell, as you know, so-called central dogma in biology. And in a cell, you know, all the proteins are encoded by the genes, which are double-stranded DNA pieces. And as you know, DNA transfers this code to messenger RNAs. And messenger RNAs have this message for protein synthesis. And with this message on messenger RNA, protein synthesis occurs on ribosomes here. Maybe I can turn on the laser pointer. On ribosomes, and after ribosome, the protein is synthesized, and we know this is the prime protein structure. And after protein is translated on ribosome, it immediately forms its function 3D structure, which is known as protein folding. And protein folding, or forming protein 3D structure, driven by some intramolecular forces like between charge residues or hydrogen bonding, and also van der Waals forces. And we know this happens with these intermolecular forces, but we don't actually know how it happens. And as you know, there are some attempts to predict this structure folding from sequence, direct sequence. And lastly, you know, the AlphaFold has gained a very big success on this, from predicting structure from sequence. And AlphaFold 2, the group of DeepMind from Google, predicted structure like higher than 90% confidence. And so why knowing the structure is that important? Because like in central dogma in biology, a central dogma in structural biology means sequence defines structure, and the structure defines function. If we know the structure of a protein, we mostly know the function of that protein. For the experimental determination of protein structure, there are three main methods. These are the NMR, nuclear magnetic resonance spectroscopy, X-ray crystallography, and electron microscopy. And as you see from the graph, X-ray crystallography main method for determining structure, but now it's reaching saturation. And electron microscopy is becoming a fashionable and technique and main technique because of the developments of in EM field. Depending on the molecule or object that you want to investigate, there are some options. There are some methods that you can use. For example, if you want to investigate small molecules, NMR spectroscopy or X-ray crystallography is the perfect method. But when it comes to proteins, NMR spectroscopy is a little bit limited, and X-ray crystallography is applied to many sizes of proteins, like to small protein and also large protein. And now we can use also single particle cryo-electron microscopy for even small proteins and also large protein-like structure like viruses or vesicles. And when it comes to cells and organelles, you can use cryo-electron tomography. And as you know, if you want to observe larger cells like eukaryotic cells, you can use light microscopy. This is the graph for all the structures deposited in the Protein Databank. It's now chosen from 2020, but it's now reaching 185,000 structures in the PDB, and it's dramatically increasing. And this share is still same. If you look at the advantages and disadvantages of these techniques, like start with NMR. In NMR, the advantage, we can directly use the protein solution. We can measure the NMR data. We can get the NMR data directly from protein solution, and that gives some advantages to us because we can get the protein dynamic information using NMR. But the disadvantage of NMR is you can use this technique in limited samples. Limited means you can only use NMR for small proteins. There is a limit to use NMR for protein structure determination. Because data processing could be very complex when you have a very big protein samples. And when it comes to X-ray crystallography, in X-ray crystallography, the main advantage, you can get very high resolution and have a very large range of protein. You can use for small protein, even big protein, like also even bigger protein like viruses. And it is perfect method for determining or studying ligand structures. The main disadvantage, the negative point of X-ray crystallography, you to crystallize your protein first. And also sometimes data processing might be very difficult because there is a phasing problem in X-ray crystallography, and sometimes solving this problem could be very difficult. And when we come to cryo-EM, the main advantage of cryo-EM, you can work with like challenging proteins that you cannot work with X-ray crystallography and NMR, like membrane proteins. And also, you don't need that much of protein with cryo-EM. Low concentration of sample is enough. And the main disadvantages of cryo-EM is not the high resolution, always not the case for cryo-EM. You cannot reach the high resolution at all, like X-ray crystallography. And also, it's not easy to access these facilities. We don't have cryo-EMs everywhere, and also the machine itself is expensive, and also maintenance of cryo-EM machines are costly. Let's start with the first method, the main method, X-ray crystallography. This is the methodology for X-ray crystallography, protein crystallography project. You start with initial bioinformatics investigation to determine what gene, what protein you want to work on. Then you do normal molecular cloning, and then choose expression system like bacteria, yeast, or mammalian, anything. Then you express your protein, you purify it, and the main thing in crystallography, you need to crystallize your protein. After crystallizing the protein, you get diffraction data from these crystals by exposing X-rays on it, and collect data, this kind of diffraction patterns, and process this diffraction pattern data, and you get the structure. Between the data collection and getting structure, there is a process called phasing, and which could be problematic. Then if you do the phasing step, then you can get the structure immediately. So how the protein crystallize, how the protein still form? We actually slowly precipitate the protein adding the high salt and precipitating agents into the solution. There are three methods for protein crystallization, and these are hanging drop method, sitting drop method, and batch method. And the main thing in these two methods is the vapor diffusion method. This is like a batch crystallization of small chemicals. And after setting these experiments, and you can see beautiful crystals like in a couple of hours or days or months. And sometimes you cannot see crystals. It's also possible. It's not predictable. So after getting your crystal, how do we collect the X-ray data? And this in this picture, you see a lab scale benchtop X-ray diffractometer. First, you have an X-ray source, like in lab scale X-ray diffractometer, we have rotating anode. And this anode could be some metal, usually copper. And with this, it's also called X-ray tube. We have X-ray generated. Then we expose these X-rays onto our crystal, and crystal is placed on a part like a goniometer, and it is used for turning crystal on all axes like this to get the diffraction data from all axes. And this is the video microscope to see the crystal on the goniometer and to align it and also to see it. We usually have a cryo cooler like to use. This is used for blowing the liquid nitrogen vapor at all times, because after harvesting the crystal, we freeze them, and we always keep them in cold, like in liquid temperature. And after diffracting the crystal, we collect the data on a CC detector. This is the close view of the goniometer, and this is the crystal on a loop, and this is the way X-rays are coming. This is the camera, and this is the liquid vapor part, liquid nitrogen vapor part, and this is the detector. And in this picture, you see a crystal in these plastic loops, nylon loops, and it's the micron of crystal. When we look at the X sources, in the previous slide, you have seen an X-ray tube rotating anode. When we compare the brightness of these X-ray sources, for example, X-ray tube has 10 to 12 brightness. We can define brightness here, photons per unit time and per unit area. When we look at the synchrotron sources, like a high-tech X-ray sources, it has brightness 10 to 25. But when we come to free electron laser sources, that you can get the dynamic information from your protein crystal, you have brightness of 35 order. Scientists usually do X-ray crystal experiment now at synchrotron sources. In the synchrotron source, an X-ray is generated by accelerating a particle in oblique magnets like this. This magnet diameter could be like several meters, like 10 meters to several meters, like 100 meters. And in these magnets, particles are accelerated. Any particle, for our case, it's the negatively charged electrons. And we use X-rays coming from these synchrotron sources. And there are like many beamline experimental stations in these big national facilities. Then you can do any experiment, not only X-ray crystallography, in these facilities. And there are many synchrotron sources in European countries, and they are all available for all scientists from all over the world, and it's free. if you look at the X-ray crystallography data collection, I cannot. Okay, yeah. When you strike the X-ray on a crystal, like always gradually return, then you can get these kind of different patterns. Like you collect these images 100 or 1,000 times, and these diffractions occur by a law called Bragg's law. It's discovered by William L. Bragg, and he got the Nobel Prize in Physics in 1950 with his father. And these diffractions occur according to this law. You have a crystal. It could be a protein crystal or a small molecule crystal. It occurs by kind of law, and where we collect these diffraction pattern data to get a structure. When I said the phase problem in crystallography, I was talking about this phase information. Because when we collect the diffraction data, we collect only the intensities, actually, and these intensities are related to the location of the atoms in crystal, but we don't know the phase information. Phase information is actually lost during this process, during the data collection. And we collect the data in reciprocal space, but we want to get the data in real space, which is actually electron dense map. Electron density map is the inverse Fourier transform of these intensities, and also it's related to their location, like X, Y, Z. But we know the phase information, but we can determine these phases using different methods. These are the main methods for in crystallography to determine the phase. They are called SAD, MED, and molecular replacement. I will explain. Let's start with molecular replacement. In molecular replacement method, you use a known structure in the PDB, but you need to look for a similarity with your protein that you want to solve the structure. There must be at least like 20% similarity. And you can also use this predicted models now. And now you can directly use the AlphaFold models because it's very successful. And you can do molecular replacement with your AlphaFold model and solve the structure data. And it's the perfect method for ligand-bound structure because you already know the structure and you want to know the structure with ligand bound. Other methods, SAD, is called single wavelength anomalous dispersion. That means you do the experiment using a one wavelength. And in these two methods, actually, SAD and MED, the other one is the multiple wavelength anomalous dispersion. You integrate a heavy metal in your protein. A heavy metal derivative of protein is produced by soaking in a crystal or by just co-crystallizing your metal. And in SAD, diffraction experiments are performed at the heavy metal specific wavelength. This is called the absorption edge. In multiple wavelength anomalous dispersion method, you can use different wavelengths. Actually, if you have metal, different various metals, more than one in your protein already, it could be native also, then you can use different absorption edges to solve this phase problem. But also, you can do experiment around absorption edge than one wavelength to solve this problem also. After getting the electron density like this, electron density are shown like the chicken wire like this, and these data processed using some software like ZL2, XDSL, SmartStream, is actually very automated at the moment. And after that, you try to fit a model in your density actually by doing this phasing operation that I mentioned in the previous slide. And you use different programs for these purpose also, like CCP4 and Phoenix. And then after that, you refine your structure like in all structure solving methods. Crystallography is a powerful tool that I will explain with this example from my PhD. For example, you see as a structure of acetone carboxylase here. And this enzyme is converting acetone and carboxylate acetone using ATP and converting it to acetoacetate. Acetoacetate is metabolized in bacteria. For example, we knew that protein already contained 3 subunits, but we didn't know how it's located, these subunits. And after getting the structure, we determine how these units are located in the 3D space. And also we knew the protein binding ATP, and also protein had like active site, metal active site containing manganese, but we didn't know where these subunits were. After the structure, we got the information ATP binding site in the beta subunit and manganese active site in the alpha subunit. And we saw the manganese in the active site. It's coordinated with 2 histidine and 1 aspartate. We even saw acetate in the manganese active site, which we interpreted as a product analog. And we also saw on the ATP binding site an AMP, and hydrolyzed form of ATP, like you see in this reaction. And also we saw the interact residues on in the ATP binding site. Also in another subunit called the CLR1 gamma subunit, we saw a zinc binding site coordinated for cysteine. These kind of information you can get only after getting the structure of the protein. That's why protein crystallography or any structure biology method is powerful to get insights into protein function. We also saw some structural conformational changes. By the way, I can say the only disadvantage of X-ray crystallography is you cannot get the dynamic information from your protein because you freeze the crystal and you get only one state of the protein. But you can get this information by getting information from different states of the protein. For example, like in this case, in the APO for the protein crystal, APO means it didn't bind anything. We saw the real conformation of the protein. But when it's bound to AMP, we saw a very large conformational change in the beta subunit. And then also there was a channel formed between the alpha subunit and this beta subunit. And we thought that this channel is used for the substrate, activated substrate, to travel from ATP subunit to active site, manganese subunit, ATP binding site to active site, manganese binding site. Because we know the substrates are activated phosphorylated on the ATP binding site. They travel from the after phosphorylation from beta subunit to manganese subunit that we got from the difference of the enzyme. Like you see in this animation, first in the apo state, the ATP binds, then it hydrolyzes the IMP. Lectins, substrate are phosphorylated, so it travels from the beta subunit to manganese subunit, and they react at the manganese active site, then acetoacetate product is formed. After explaining the X-ray crystallography, we can directly go to nuclear magnetic resonance spectroscopy, where you can get dynamic information, not like X-ray crystallography, because you use your protein solution, then you get these dynamic information from your protein solution, like in seconds, in minutes. In nuclear magnetic resonance spectroscopy, local magnetic fields are observed around the atomic nuclei. The sample is placed in a magnetic field. Here is the cold environment, the liquid helium, and liquid helium is cold because of the liquid hydrogen around it. And the sample, the NMR signal is produced by excitation of the nuclei sample with radio waves into nuclear magnetic resonance, and which is detected with the sensitive radio receivers. And these nuclear magnetic resonance signal information about the relative bond distances and angles. These relative bond distances and angles have information for structure of a protein, as you know. So how do we do protein structure determination by NMR? We use experimental structural restraints like nuclear overhauser effect, NOE, Dihedral Angles restraint, and hydrogen bond and amide protein proton exchange rate restraint. Let's start with the explaining the nuclear overhauser effect. NOE is the intensity of nuclear resonance with paramagnetic nuclei. That's why we need to either topically label protein. NOE enhancement between nuclei are caused by dipole-dipole interaction and inversely correlated with internuclear distance. By using this information, NOEs between neighboring residues, we can get the secondary structure information of a protein. And also, we have other information called dihedral angles we get from J coupling constant. These dihedral angles are the phi and psi angles on a peptide bond. That one is the psi angles between alpha carbon and the other carbon, and the phi angle is the alpha carbon and nitrogen. And these angles are actually change between in a certain range, and not possible to be in another range actually. And we know these values, and they are also known as a Ramachandran. And the Ramachandran plot is graphs against phi angles and psi angles, and we use these restraints actually to refine any NMR structure or any X-ray structure also. And after that, we have another restraint, is the hydrogen bond restraint, getting from the amide proton exchange rate. Amide protons that are protected by hydrogen bonds or other interaction with hydrophobic residues tend to show lower exchange rate. Then we can get different hydrogen bonding information from these residues in the secondary structure of the protein. And after using all these strain, NOE strains from neighboring residues, Dihedral Angles restraint, and hydrogen bond restraint from amide protein exchange rate, we get the final structure using these NMR strain and using NMR. When we come to this graph again, the NMR EM X-ray, the graphs for structure determination, we see that 2014 is the date that the cryo-EM has accelerated a little bit, and we know this date, 2014, is the cryo-EM resolution revolution, because in 2014, in a article in Science, Werner Kühlbrand said 2014, the beginning of a new era in molecular biology, where structured at near atomic resolution no longer the prerogative of X-ray crystallography or nuclear magnetic resonance spectroscopy. Because he knew that because after the revolution in the field, like in software or in detector system, he knew the resolution will be higher after that date, and he was right. After 2014, after this article, cryo-EM was mentioned in Nature in 2015 as a method of the year, and also Nature methods. And this method received a Nobel Prize in Chemistry in 2017. And recently in 2020 and 2021, we got even like atomic resolution using cryo-EM, like 1.2 angstrom, 1 angstrom. We could even see the hydrogens in the protein structure, like in this picture. So why the resolution is that important? When you see this picture, maybe some people have already know. You have some ideas in your head, but you don't know what it is. But when you see when you use smaller pixels, when you use higher resolution, then you can see what is in the picture really, actually. Then you can see it's an all. That's why the resolution is that important. Because the cryo-EM wasn't a resolution method in all time. For example, in 2014, when the resolution revolution had started, the resolution average was around 20 angstrom. Then it dramatically decreased. Then actually the resolution as a number decreased, but it's actually increased with high resolution, low number. In 2019, the resolution average was 5.6 angstrom, and with these high resolution structure, and 2021 maybe it's a little higher to 6.9, but it's still high. But for example, we look at the high resolution structure. For example, in 2016, we have 1.8 angstrom resolution structure, which is an enzyme glutamate dehydrogenase. And when we look at the 2020, 0.1 angstrom, like 1.15 angstrom resolution, apophoritin has been solved. Now that we can see the atoms like individually, even hydrogen. That's why because of the developments in the previous couple of years, this method, cryo-EM, got the Nobel Prize in 2017 in Nobel Prize in Chemistry, and shared with 3 people. Jacques Dubochet, which developed the sample preparation with the freezing of the samples, and Richard Henderson and Joachim Frank, which who developed the sample optimization and data acquisition and algorithms for 3D reconstruction of the models. Let's see how cryo-EM samples are prepared. Actually, we put the, when we prepare the cryo-EM sample, we put the samples, like protein samples, on a grid. The grids are metal mesh like this, like 3 millimeter diameter metal mesh, very thin, and they have a very thin, like a nanometer the thickness carbon film, and this carbon film has some holes like this. And after putting the protein sample on it, when we get the excess amount of protein sample, then after that we immediately freeze the grid in the liquid ethane. This is the liquid ethane, and this is the grid on the tweezer. And this is the liquid ethane tweezer and the sample on grid, and a very thin layer of solution, and it's immediately frozen in the liquid ethane, and around it there is a liquid nitrogen. And this is another picture that we can see the particles and grid. This is grid mesh, 3 millimeter, like made of metal, like usually copper, and could be molybdenum, nickel, or gold. There is a very thin layer of carbon nanofilm, and this carbon nanofilm has holes on the film, and actually the protein particles, protein molecules are located in these holes, and around the protein particles there is a very thin layer of amorphous or vitreous ice, and these are the embedded particles in the ice. And there's a very nice video here to explain what is happening in electron microscopy. Okay, when we have the sample, like very dilute and pure sample, you see the individual particles in your solution. And when you freeze them, like immediately in liquid ethane around minus 180 degrees Celsius, and you get your protein almost the native state, like individually in amorphous ice. When you transmit the electrons onto this sample, then you get some from your protein sample, from your particles, which are at different orientation in your sample. Because they are at different orientation, then you can get different two-dimensional pictures from your protein, actually. Then but also you get these pictures, you take these pictures like for example thousands or millions of times, and at the end you have at least thousands or up to millions of pictures. Then you combine all these pictures to get a like 3D dimensional picture of your protein, of your cell. Okay, this is another way to look at this process. You have this particle on your grid. This is a negative chain micrograph. After negative chain micrograph, you freeze your sample. And after freezing sample and taking the picture micrograph on cryo-EM machine, you see your particles like this dark gray one. And background is your solution, like amorphous ice. After that, you pick particles. Then you can use also a program to do that, actually. And you classify these pictures, and we call them 2D class average. And after 2D class averaging, class these 2D class averages in a 3D classes, with a less number of 2D class averages, obviously. Then after that, we continue with one 3D class. Then, we got a refined model at the end. And in the refined model, we can see we get the electron density map, the electron microscopy map, that in which we can see all the polypeptide chain and also details the side chain like phenylalanine, isoleucine, if you have a high resolution map. Actually, we do this using softwares. One of them is the most famous one is Relion. We also use this program. In Relion, we get the electron density, electron microscopy map that gives protein model to you by starting from the raw data. Raw data is the micrograph that you collect from the aquarium machine. When we summarize all the structure methods in this graph, according to resolution, accessible size, speed of expansion, we know all these methods, NMR, QAM, we explained, micromolecular crystallography, high resolution method, but they require some time. But there's another method, it's very easy, very convenient, called SAX method, small angle X-ray scattering, and use for any protein, any kind of protein, small protein, large protein, and you can get information like in a minute, in hours, from your protein. This is because this is also a solution technique, and you don't need to either to label it like in NMR. And from SAX, small angle X-ray scattering, you can get information about size, oligomeric state, and shape. And because scattering intensity of X-ray decreases, it's like X-ray crystallography. The only difference is we use solution. The intensity exponentially decreases by increasing angle. We use a small angle. That's why it's called small angle X-ray scattering. And we got some graphs. And for example, the most important one, the intensity in the center, that gives information about the molecular weight and size. We can also identify the foldedness or unfoldedness of our protein, but just in minutes using some graphs like this. And SAX advantages is the rapid data collection and processing. Can assist in understanding biological structure and function. It is used for validation of crystal structures. If you have a multi-subunit or multi-protein complex, you can insert these crystal structure in your SAX model, for example. And the main advantage is that you have to add isotopic labels like in NMR, or you don't need crystal diffracting crystal like macromolecular crystallography. And now structural biologists use combine all the methods, all the information from different methods, not only the main methods like crystallography, NMR, electron microscopy. We also combine the information from SAX, like I mentioned, like cross-linking mass spectrometry, too much information about especially for protein complexes, proteomics, like different biological strategies like co-purification, and also bioinformatics data. We combine all these information, all these components to get a final model, final outcome. This is called actually integrative structural biology. So how is the structural biology in Turkey? And we believe in the power of integrative structural biology, and we came together and we established our structural biology community, and we aim to reach young scientists interested in this field. And we're a small community actually, but we're specialized in different fields in structural biology, from computational structural biology to SAX, cryo-EM, biological NMR, serial protein crystallography, and protein crystallography cryo-EM. All the methods we are specialized, and we aim to reach you and young scientists who are interested in this field. That's why we organize workshops. Like we just organized in early November the second National Structural Biology workshop, actually face-to-face in Izmir, and it was partly also virtual. But we also organized our first National Structural Biology workshop was in February last year, and all videos from that workshop are online on our YouTube channel. It's the Yapısal Biyoloji Tolu or Structural Biology Committee. You can find it on YouTube, and all the videos are on YouTube, but they are all Turkish actually. But you can benefit from these videos, and you can subscribe our YouTube channel. This is the end of my presentation, and I would like to thank firstly TÜBİTAK for giving this opportunity to do structural biology research in Turkey. Also, I will like to thank my team, BBK Lab, to realize structural biology research in our lab. And I got my crystallography training at Imperial College London in James B. Lab, and I would like to thank him also. We had a collaborator for acetylene carboxylase project, Will Diamond, micromolecular beamline, and I got my cryo-EM training in University of Bristol at Christian Berger, Schaffestell, and Imre Berger lab. And we used cryo-EM facility at Bristol, and also we used Diamond EBIC facilities. And thank you for your attention, and I'm happy to take your question. Thank you.
Thank you very much for your very nice and clear presentation. If you have any questions, you can type your questions into the chat box.
Can I?
We have one question regarding to protein structure determination by NMR. What do people normally concern about when amide proton has higher exchange rates? Does that mean we have lower confidence on the structure formed at the particular part of the protein? Thank you.
Okay, thank you for the question. Actually, firstly, I'm not an NMR person. My specialization is not NMR, but higher exchange rate. Yeah, it's, the amide proton has higher exchange rate. Actually, this is the I realized this new method, like a residual dipolar couplings as well. I didn't mention that. Yeah, I don't know actually. I'm not expert to answer this question, but I think you get some kind of information from about the hydrogen bonding from this kind of experiment.
We have another question. Is there any kind of internship opportunity being offered by your institute for hands-on practice?
Internship, yeah, we have internship opportunities and also provided by TÜBİTAK as well. We always get interns all the time in our lab. But I'm not sure if it is available to the international students. But yeah, if one can do like voluntarily, then we can arrange something, I think. Yeah.
Another question is, for structure modeling, if X-ray structure is not available, so which structure and measure will choose for the selection of targets?
For selection of target means like the protein that you want to solve the structure, or like a drug or small molecule here. Not available. Which structure measure which is for the selection of target? Protein. Okay. Yeah. I mean, there are limited techniques that you can solve this the protein structure in atomic detail. There are actually these methods that I explained actually. But if you don't have access to X-ray crystallography, you need to benefit from other techniques. But it is limited. If, for example, NMR. For NMR, you cannot use NMR for like very large proteins. If you have a protein like a very large molecular weight, then the only option there's a cryo-EM and X-ray crystallography. Also, yeah, I think these are the techniques that you can solve the structure atomic detail. Yeah, I would choose the cryo-EM if it's protein other than X-ray crystallography. But according to accessing possibility, I think X-ray crystallography much easier than cryo-EM to access.
Another question is, given the concentration of protein and precipitant, how can we calculate the rate of protein supersaturation?
Protein precipitant, how do we protein supersaturation? Yeah, actually, there are some graphs. There are some to show the protein precipitation by the concentration of precipitant. But we don't usually calculate this actually. Because now there are like numerous conditions that we can use for crystallizing protein, and it's also commercially available. We use them, and we just hope our protein will crystallize. And there are some studies about these, like calculating this the supersaturation or precipitating kinetics. Yeah, but we don't do like protein crystallographer or structural biologists don't usually do that.
Another question is for cryo-EM during sample preparation, if the protein is non-soluble protein, can you share a brief description?
Protein is non-soluble protein. Then you're in trouble, I think. I think you need to find ways to solubilize your protein, to increase solubility of your proteins first. I guess because cryo-EM, you need to you're looking for like a single soluble particles of your protein in your solution. I think you need to find the strategies for the solubilizing your protein. For example, changing buffer to pH, or for example, SAX is the perfect method to investigate these conditions, because you can get like information like in minutes and in seconds from SAX, and you can investigate this condition at which your protein is the most stable. And there is another method called CD spectroscopy, circular dichroism spectroscopy. You can also get information about folding of your protein, stability of your protein, and then after determining these conditions, then you can use the higher technology structure determination techniques.
Another question is how to choose and validate a protein from protein database in Silico, and if at all some amino acid residue are missing from PDB structure, how it can be rebuilt.
How to choose and validating database. In Silico, okay, how to choose and validate protein. Actually, this validation made these NMR restraint. X-ray crystallographers also use these NMR restraint to validate structure, and also there are some online servers to these validation. Like the first one is called Molprobity. I can get here. This is made by using the NMR restraint, and this validates the structure according to the Ramachandran plots, like rotamer outliers, according to clashes and Ramachandran outliers. Then you can validate any structure, any PDB using these kind of servers, this kind of software. For the missing parts, in principle, if you don't see any data from like your experimental data from your experiment, you should not model any protein structure over there because you don't see it. But you can maybe predict it because you know the sequence, and you can model it if you really need it. Or also maybe you can use another data. You can use this model, like the model with missing parts, and do another experiment to complete these missing parts. But there are also other software to do this. For example, the main software we use, Qt, to do our modeling on our experimental data.
Thank you very much for your answers. We have some other questions, but because of the time limitation, I need to stop.
I can run chat.
Thank you very much. And thank you again for your nice presentation.
Thank you.
And the next session is the practical class will be given by Cemil Can Saylan about important websites and visualization with PyMol. Now we have a break. See you in a few minutes. Hello again. And now we are ready to move on to next session. Next session will be an hands-on session. It will be given by Cemil Can Saylan. And in this hands-on session, you will see the important website about structural biology and visualization with PyMol. Cemil, stage is yours.
Thank you very much. Can you stop your screen? Okay. Okay. First of all, let me introduce myself. I am Cemil Can Saylan, and I am a master's student in Computational Science and Engineering Department at Istanbul Technical University in Turkey. And I'm working on molecular modeling, simulation, and also computational drug design fields. And today, together, we will see some database and some visualization of some structure in PyMol, and also I will show some brief visualization in the VMD. And also, first of all, actually, I would like to thank you joining for this workshop. Before we start, let me show our GitHub page. After this workshop, we share some information about the in this workshop in this GitHub page. I put in this link in the chat box. You can reach, and in here in the useful website and visualization part, I put here a table. I take from this paper in here. You can see this comprehensive database and the websites. Today we will cover just 4 of these websites and databases, because we cannot I cannot show all these databases in this workshop, as you can see. You can read much more detail in this paper about this website. And also after the workshop, you can try and you can gain the experience using this tutorial in here. I put this official tutorial PDF in here. And before, if you didn't download or install the PyMol and VMD, you can use these links in here. And also in the PyMol, you may register the education license in here. That's all information now I need to tell you. Okay, let's start with the most known database, which is the Protein Databank, actually. Protein Databank, it is a worldwide known repository, and it contains all necessary information and all all structural structure in the biology. I mean, all the structures coming from the different techniques that the previously you see in the theoretical lecture. And this database coordinated by Protein Databank, Worldwide Protein Databank Foundation, and they also they also provide these databases. And also, I would like to mention this PDB 101 because this is something like educational portal of the PDB databank, and I will give you some introduction for this database, but you can read in here different information, like let's say right introduction in here. You can say that what is the protein. If you don't anything about biology, you can read it. But let's say PDB, let's say PDB format, for example. And there's a bunch of different introduction and bunch of different information in here, and some of them are very detailed, but and also you can find the very brief introduction of the structural biology. This is maybe the if you don't have any experience in the structural biology, maybe this is the first step for you to jump into the structural bioinformatics or structural biology. Let's continue with the Protein Databank. And in the Protein Databank, first we will see this web page, and you will see the latest entries, feature and highlights, and also news in the Word file news in here. And we searched in the database different searching options in this database. First, actually you can search with the keywords and also use the PDB ID. These PDB IDs are unique 4-digit codes for the structures. These all unique for these proteins. For example, if you write this code in here, you will see that this structure, and this is unique. This only represents this structure. Let's say, first we search the keyword with the keyword. If you write the SARS-CoV, it brings you all structures related with this SARS-CoV keyword. As you can see, it's give the all structure, and you can filter or refine this structure in the left panel in here. For example, you can select the Homo sapiens, or you can select which experimental method you want. Let's continue with the X-ray diffraction, for example, and you will get the all structure with this filter. And also you can search with advanced searching query builder. And in here, you can give the sequence of your related protein or your desired structure or sequence. Or also you can search with the chemical attribute. There is different chemical attribute in here. You can use you can search with the directly, or you can search to your ligand that's found in the different structure or proteins. And also you can search with the PDB ID. Let's write a 3FAD. And as you can see, we only found just one PDB because this is for this protein. Let's open this one. By the way, I shared the PDB files that will be examined later in the PyMol parts. You can download in here this data if you are not access the, or you cannot, if you cannot download it in the PDB. Okay. When you open this PDB structure, one of the PDB structure in the PDB databank, you will see the nice presentation, nice image of the protein, and you will have the very brief information about this protein. And this is the lysosome with mutant at the 96 arginine to histidine, a point mutation in here. And you will see the organism and expression system for this protein. And also we see its method and also the resolution of this structure. And PDB also provides us its literature information where it can, this structure were purified or where they get this structure. And also they give this PubMed and its DOI ID or DOI ID. If you click this one, you can directly reach to the literature information for this structure. And also we see the macromolecule content in here. They give us its detailed information about the structure. For example, we see the atom count and also the total structural weight of this structure. And also very nice things in the PDB, you can see what inside this structure. For example, in here we see that there's only one chain, which is the A chain, and its length, sequence length in here. Also, they provide its uniport ID, and if you don't know the uniport, you can click and you can check. I'm not going to show the uniport in this workshop, but you can search. In the uniport you can find much more detail about this protein, and not direct the structure, but the protein. In the protein feature view, they also give the other, they actually combine different tools and different websites in here. For example, you can find the disordered region. You can see the disordered region about for this protein. And this came from the IUPRET2 short. You can see in here when I came here, as you can see. And this IUPRET gives the disordered region scores, kind of scores for the residue by residue. And also we have the PFAM, which gives the domain information for this protein. And also we can take the we can get the small molecules in this protein or the structure. For example, in the structure we have phosphate ion and also beta mercaptobetanol, something like that, in the structure. And also we PDB provide a brief description of the experimental validation and the data set. This is much more actually complicated if you are not familiar the experimental procedure or if you don't know the experimental stuff, experimental procedure actually. And also it's the entry history. And let me come here. There's very important things that you need to know in the structural biology and also in the PDB. PDB represents structure as either asymmetric unit and also biological assembly. There's a very distinguished, there's a very unit, there is a basic difference between this asymmetric unit and biological assembly. Asymmetric unit is a minimal representation of the structure in the crystal, and also let me show you in here actually in the PD 101. Let me see asymmetric unit, for example. Yeah, biological assembly and asymmetric unit, and you can see they provide this asymmetric unit. But when you came here, you can see the entire crystal actually, and this is what we see in the actually unique part of the crystal. Actually, in the this asymmetric unit is the unique part of the crystal. What is the biological assembly? This is actually the functional complex of the structure. Functional complex of the biology. We thought that this form is the active form, or that this form is the native form. It's found in the biology. For example, in here, this protein can be worked as a monomer by authors. They assigned by the authors. They give the information here, and also they found out that this protein can be worked as a dimer. In this information generated by the software, which is the PISA. And to understand this, let me give you another example, which is the true bug in here. For example, this is the satellite tobacco necrosis virus, and this is also produced from the X-ray diffraction method, and they obtain this asymmetric unit in here. But they all know this protein or this virus looks like this, and they generate this biological assembly from this asymmetric unit. And in the Protein Databank, you can download this structure for the structure with different files format. For example, you can download its sequence with the FASTA format. You can download the PDB format. You can download its GIF format or this format, and you can download the biological assembly. I will give you. Actually, I gave you this structure in here. We will see in the primal section, and you can download. And also, we can download from here. For example, please download if you didn't download a PDB format, or also this 2FOFC map. This is the X-ray diffraction density map for this protein. These all calculation for density map. I will show you this 2FOFC map in the panel. This is the X-ray method. Let's see another method. For example, let's write CNC0. This is the one of the potassium ion channel. This is also called this HERG channel. This is this structure produced by the electron microscopy, and its resolution is 0.90. And the inside of this, actually the summary of this structure is similar to X-ray crystallography and also the other methods. And in here, we see the structure summary of this structure, and also you can see other information in here. For example, when you came here in genome, if, for example, they didn't assign any information for genomic part, let's look. Let's come back to for 3FAT. When you came here in genome part, they also provide you this chromosome IDs and the other information in here. And also, if you came here this sequence part, you can see all the other information in here. For example, its mutagenesis, its active sites, for example, in here. They provide by the uniport. You can see the right part, upper right in this table. If you don't know anything about your active site, but there's information about the active site of the structure, you can came here to see what kind of active site information you can have, and also the binding site in here, for example. Okay, in the experimental section, they give much more detailed information about the structure in here. And okay, let me continue with this. Yeah, I forgot to mention, but in here, if you don't have any third-party software to visualize your structure, you can use there's an embedded. There's a feature embedded in the Protein Databank, and if you click this structure, you can visualize your protein in your browser. You don't need to download, or you don't need to use the other softwares. And this is the very quick overview of your protein, and you can also change the appearance of the structure using this panel. And also you can see this density file of density of this structure in here. Let me maybe you can see in here. Okay. This is for example, the density of this structure. As you can see, if you open the density in here, you can see this is a very quick overview. You may not see all features, or you may not see all detailed information about the protein, but if you can see the sequence, and if there's any missing region, maybe you can detect in here. Okay, let me continue with CN0. And as I said before, this is electron microscopy, and as also it's the membrane protein. And in the cryo microscopy structures, you will see the asymmetric unit and biological unit as a same structure, because we directly model and directly take from the asymmetric unit as a actually model as a biological assembly. The other information are the same for the cryo structure. And next, I will like to continue with the other database, which is directly related with the EM data source. And in the Protein Databank, you can reach the other website and all other useful database in here. For example, for this structure, there's a link in here. As you can see, there's a EM data resource, which is the other another database we will cover today. And if you click this one, it brings this website in here. Before we look at this one, let me show it here. This EM data resource for directly electron microscopy structures, it provides the data archiving and retrieval, news, events, and software tools, and also data standard, validation methods, and community challenges in here. You can see the recent release in the EM databank, and also, and there's a news in here, and also you can see the different tools in here, and similarly in the protein, the structure in the EM data source have the unique ID, and you can search this ID by writing in the search panel or in the homepage of this EM data source. When you search this structure, let me search, for example, this one. Look, it's directly open the same web page in here. What we see, it gives the very brief summary of this structure, and it also provides samples what was in the it gives the information about what contains in this structure. And also it gives the experimental information, which is much more detailed when we compare the PDB, actually. And they also show some other softwares. For example, when you can see the other techniques for other structure, they use the Carrot, Code, something like that, or Rosetta, or other softwares in here. And also you can download this, and also you can see the map information. And I would like to mention this contour level, because when you download this map, they suggest that the best resolution or best contour level to visualize this structure in here. I would like to show this map, but I'm not going to show you this map because it's much more larger than actually it's a larger structure actually when we compare the others, and we have limited time to see this kind of, and also you can encounter problems when you visualize. And similarly, I'm going to show you the X-ray density map, and these are the similar when it's visualized. And other things I would like to talk about the validation. And if you came here and they provide the validation report, and if you open this validation, they give you very detailed information about the structure. And you can find the model quality, model visualization. And if you came here, you can see which primary map and which image they take from their experiment, and how can combine, how can generate this model. They give the information about how they generated this model. And also they give the map analysis and also model fit information. If you work the structure bioinformatics, sometimes you need to check the structure and also density, because sometimes sometimes the software may have some problems, or you always need to check your actually project, or I mean, you always need to check your structures. in the structural bioinformatics and the other related areas. Okay, this is the EM data resource. And also they gives the other useful links in the links section. You can go directly by clicking to this Protein Databank and other the database. And as I mentioned before, this is the membrane protein. And membrane protein can be challenging because you need to define the membrane location, and you need to define the membrane composition of the structure and also this protein. And because of that, I would like to give the other website, which is the OPM. This is the orientation of the proteins in membranes. This is this database gives you the spatial arrangement of the membrane proteins. You can define or you can find the unique experimental structure of the transmembrane proteins, and also some peripheral proteins and membrane active proteins in here. You can actually let me start over in the OPM. Sometimes you need to place your membrane in the your structure, and they give some different server in here, for example, in the PPM server. And you can, by using this, you can calculate the rotation and translation position in the membrane of the transmembrane in the membrane structure. When you launch this PPM, you can see this section, and you can give the coordinate file in here, or you can search the PDB entry, or you can search the OPM entry in here. Before that, I need to tell you some about much more detail about the. Let me give you much more detail about the OPM. And they classified the structure with this format. They classified as a type. They looked whether it's transmembrane protein or monotopic peripheral protein or membrane active peptides, or they classified as all alpha, all beta, and alpha and alpha/beta, or non-regular proteins. And this is different classification when compared the taxonomically, maybe. In here, also they give the very nice information about the biological membranes. If you would like to read, you can come here and look the biological membranes, and they give all information about the other biological membranes like endoplasmic reticulum or eukaryotic plasma membranes. They gave the anatomy, and also, for example, for membrane for Golgi apparatus. And because in the structural bioinformatics, you need to know where your protein embedded in the cell. For example, it could be in the endoplasmic reticulum, or it could be in the plasma membrane. And they all different composition when we compare as a membrane. Okay, I'll talk about this PPM. Let's continue with the search. And in here, we have this 7CNO0. Let's search this protein in here. And as you can see here, they give its classification information and also its resolution. And this EM gives this structure came from the EM electron microscopy and its protein ID. And they also links to they give the links other databases. And in here, I would like to show you these transmembrane segments. They have this structure has 4 chain, and each chain has 6 transmembrane segments. And these IDs are the amino acid, or we say the residues of this protein. And this, for example, this residue is transmembrane transmembrane segments of this structure. And here they give this image, and this blue dots are the cytoplasmic sites, and this is the extracellular site. When you download this OPM, and you will see that this image, and I will show you this in the PyMol as well. Okay, this is the OPM for the membrane proteins. And lastly, I would like to talk about the AlphaFold protein structure database. This database is very new, and I assume that you all know the AlphaFold. And if you don't know, let me explain briefly. AlphaFold is a deep learning algorithm to predict the protein structure, and it's actual state-of-art method, and it's very huge. I mean, it's a very huge it brings a very huge progress in the prediction of the structural prediction of the proteins. And they collaborate with and actually the DeepMind and the EBI collaborate to build this database, and this is actual beta version. They're not released fully, and they're not released full version. And they provide the protein structure predictions for the human proteome, and also they provide the different organism proteome actually, and the other key proteins of interest and accelerate scientific research. And in here, in the download section, you can download all the structure they give in this database. And actually, in when you browse or when you search the structure, you cannot find the proteins longer than this 2,700 amino acids, and they give this structure 1,400 amino acids longs in this downloads links, and you can, and also they give the they give the structure with the unique names in here. You can read this in here. Let me search, for example, this key arc channel in the AlphaFold. Let me come back. And when you write your keyword in here, also you can search with the uniport IDs, or you can search the organism in here. Let me search. And they brings a different structure, and you can filter by the organisms. And let me click first one. Okay, and here and here we can see the structure, and this blue colors represent the high confidence in here, and this orange one is the low confidence. And if you don't know the AlphaFold, AlphaFold actually not give very well structure if you don't have the higher amount of MSA data, or let's say if you have lower amount of MSA data, which is the multiple sequence alignment, and also if your protein is interesting to the disordered region, it may give the not fully predicted structure. It maybe gives this kind of, it can be give this kind of things, but these are very huge progress actually when we think the structural prediction methods, among the structural prediction methods. These And these blue colors are high confident, as I said here. And that's why they give the prediction aligned error in here as a heat map. And in the feature, they will give the sequence feature in the next version of this database. And this is the other database I would like to say. And again, let me came here to this GitHub page, and you can see all this web page or web database in here. And today I would like to say this 4 web page and databases. And now I would like to continue with the visualization part of the this workshop. Let me check this chat box if you have. How are the given databases useful in the prediction of not yet? What? How are given databases useful in the prediction of not yet unknown protein structure? For example, in this AlphaFold, they provide the structure actually not fully released or unknown structure actually, and you can actually search to the Google to reach the collab of the AlphaFold, and you can use this collab to predict your structure maybe. I will show you the electron density map in the I showed the electron density. How can you visualize in the RSG? Okay, let's continue with the visualization. Okay, I'm using the Linux actually. If you install the PyMol, you can open and you can open if you would like to follow me. And here you can use this installation links, and if you follow this instruction, you can easily download and install the PyMol. And let me open the PyMol first. Let's go to I actually write in here as a PyMol, but you need to give the full path of the PyMol to open this one in your Linux system. And maybe if you are not using the Linux system, you can, if you use the Mac or the Windows, you can directly clicking the PyMol image or PyMol icon and to open this one. When you open the PyMol, you will see this interface, and probably if you use the PyMol first time, you will see this kind of activation pop up in here. And as I said before, if you register and install this education here, after that you can browse your license, and you can open the license, and after that you will see that this education use only text in the left part of your interface or workspace. Okay, this is the how looks like the this is the PyMol. Actually, this is the nice log file or the information about your system and which kind of the GPU, or it's give the information about your CPUs. And in the right side, you see the panel. You can use the while you are or while you are visualize your structure. And I will maybe show this. I'm going to show some of them. In here, you see the entry box in here, and they give the very brief. They provide the quick links in here. For example, this A means the action, and you apply some different action in here. And in the S, which means the show, they change the visualization of your structure, and this H means the height, and they also gives the opportunity to disappear or to some unwanted things in your structure. And you can this is the L for the label. You can label your structure or part of the structure. And this the C means the color. You can change your colors in here. And in the bottom right section, there's another panel. And in here, they provide the viewing mode and also editing mode. If you click near something near this location, they change the Pymol switched your mouse mode as a editing mode. Be careful about this because you may do some unwanted or not nice action in your protein. I will show you some quick detail in the structure. And this is the selecting. And if you click this selecting, you can select different, you can see the different options. For example, you can select atoms, you can select the residues, you can select the chains. Let's see this information on directly in the protein structure. And before that, I would like to talk some about the protein. I would like to talk about the data formats. And in here, basically, we have the PDB structure format. Let me open my data in here. When you open this PDB format in your text editors, and first of all, you will see this metadata of the structure. They give the related information about your structure. For example, it's literature, and it's, for example, about your experimental data information. And also, they give some information about your protein. And basically, this protein, this PDB data format or other data formats provide you the coordinate of your structure. And in here, when you came to this section, you will see the atom. And this is the what we call the PDB actually. And this all information is for your information. You may not use this information as well, but this atom files is the necessary for the PDB. And this is the unique structure format of the PDB. First, you give you define your atom. This is the your atom number, and this is the atom. This is your atom type, which is the nitrogen, and this is the 3-digit letter of your amino acids. And this is the chain ID, and this is the residue number of this amino acid or residue. And this is the X, Y, Z coordinate of this structure. Basically, you can think that this PDB format or other PDB or other data format as a container of the coordinates or container of the atoms. Yes, coordinate container of the atom coordinate positions. And in here, there's a occupancy and also some beta factor of this structure. This beta factor, also called the temperature factor, it's how your residue or how your protein is, how much your protein is movable or how much flexible in your structure. Okay, and in for example, other data format is the GIF format, and this gives much more detail when we compare the PDB format. It's also continues to evolve. They add other information for they give other information about the proteins. And the basic format, syntax format of this PDB. First, they give this attribute of this is the category of they give the category of and then they give the attribute of this category, and they give this information. Okay. And similarly, when you open or download the other G format, when you continue in the file, you will see the similar approach in the PDB as in the PDB. And when you see the atom in here, it starts to give the coordinate file of these atoms. And you can see these attributes and also labels for this structure. Okay. If you would like to read more about this data format, you can came to this PDB 101 and you can read this this blocks maybe. Okay, when you if you would like to open a structure, first you need to do is came here to this file, and by clicking this open, and in here let me open this 3-bug first. And as I said before, let me came here to 2-bug. As I said before, there's a asymmetric unit and biological unit. When you download this PDF format in here, and also in the biological assembly in here, if you download these two, please download these two actually. And when you open this PDB in here, you will see this kind of the image first. And there's a spheres and also the helix and beta sheet. These the parallel ones are beta sheet, and these are the alpha helix structures. And there's these red dots are the oxygen. And how can I know? Actually, I have experience, but if you don't know, you can come here, this the right bottom. There's a S ladder in here. When you click this one, it's open this panel in here, and you can see the one letter of this residue's name. And also you can, if you came here, there's this O letter in here. This means that these are the water, oxygen in here. And in the X-ray crystallography, you cannot get the hydrogen atom. And some of them maybe gives the hydrogen atom information, but most of the structure cannot give, actually not provide the hydrogen information. And also in the PyMol, you can see this oxygen like this. Okay. And I would like to show this biological assembly as well. Before that, we need to change this name entry in here. Okay, in the action here, you can, if you click this action, and you can see the other function you can apply this protein. You can rename the object, or you can copy the object to a new group, new object, or you can group the other object within the you can group the other object with the new group. Okay, let me rename it first. Let me say this 1. And if you open the biological assembly, when you download, it's give this compressed file PDB1. And in the PyMol, you not need to uncompress this file. You can directly open in the PyMol in here. When you open this file, you see that the second sequence is appended in this panel. And sometimes you need to open the large, too many objects, And if you and in that case, you will see all the structure. But if you don't want to see this one, you can close or hide this structure by clicking this object file, and it will be hide in your workspace. And here we download, but we couldn't see the whole biological assembly in here. But if you came here to this, actually it's written here as it's written in here 1/60, which means that this structure or this object has different states or different trajectories. You can see these trajectories in this right panel by clicking these arrows, and you can see these are the different states of the protein in here. For example, I actually changed the way of looking this one. And if when you came here, for example, if you click the reset button in here, it's reset this structure. If you make trouble when you are visualizing, you can reset or zoom orient options to start your visualization over again. And when you click these arrows, you see the different states. But I would like to see the whole biological assembly. How can I do that? When you came here, this action, and there's a state function state options in here. When you came here, and you can see the all. If you click the all state, you will see the all state of this structure. And as you can see here, this is similar to this our biological assembly. Now we see all this biological assembly. Also, you can split these states with individual objects, but I'm not going to do that because we have too many states in here, like 60. Okay. Let me close this one. Let me continue with the other structure, which is the tree fat. When you open this one, we get similar structure. Okay. As you can see here, these oxygen atoms are kind of noisy. When I look at this structure, I would like to get rid of this oxygen first when I visualize. What can I do? When I came here to this height section, and when in the height section, you can see that there's waters. When I click this waters, there's it's directly hide these waters in the structure. And also there are different heteroatoms in here. And when you in the selection of the PyMol, you can select your residue or atoms or other things in your structure by clicking like that. And when you click, it gives you the different, like this. When you click, it shows this kind of things. When you show when you click this things or this heteroatom, and it shows this kind of image to represent that you choose this heteroatom. Or you can choose in this sequence panel in here, or you can choose the one or more sequence, like choosing this one, or you can use this comment line in here. In the PyMol, you can use different comment scripts, and sometimes you cannot make what you want, or you cannot do what you want in the this interface or the quick links, and you need to write some codes for the structure. For example, there's a code named select, and you need to define your selection criteria, for example, res name or residue. And you can find actually this information in the PyMol wiki. There's a kind of blocks in here. And there's a tutorial file, and also you can reach the other scripting of new plugin about for the PyMol. And okay, for example, when we select this tyrosine in here, and if you want to directly focus this tyrosine, and also before that, I need to mention when you're selecting any residue or other chain or other kind of heteroatoms, the PyMol creates a new object which name is the cell, and if you do operations in this cell, it's directly applied for this selection. But if you apply the visualization, it's directly applied the whole structure of this protein. For example, there's a different representation. In the representation part, there's a difference. For example, if you select, if you show S, it's apply whole structure. It's change whole structure. For example, let's click show and S, and if you select the licorice, it's change whole structure as a licorice. But if you select, if you change your structure representation without the S, or if you use this selection, if append your visualization directly to your your workspace, and you will see the both of this representation. And let me make this cartoon. For example, let's make this. Let's append the surface in here, and you can see the surface representation. But we also need to know. We also should see the cartoon, but it's behind this surface. I will make it visible. I will show the how can we visualize this cartoon. But before that, I would like to change this surface color in the colors part of this color option in here. And there's different color methods in here. You can color by the elements. For example, if you select this one, it means that the carbon atoms will green, hydrogen atoms will be the white, and nitrogens are blue. And it gives this kind of structure, this kind of representation. And for example, we can visualize by chain. Also, you can visualize by the secondary structure. For example, when you click this one, these red ones are helix, and beta sheets are yellow, and the loops region are represented as green. Okay, but you can also show your coloring by looking at this B factor. When you're clicking this B factor, it shows where your structure more rigid or where the more flexible in here, as you can see. And also you can select also you can show your structure as a rigid color. For example, let's select the white in here, as you can see. Okay, and I would like to show other things. For example, in here, Pymol provide you settings and also display display option. You can change your workspace visualization, and also you can change you can change the appearance of your surface, cartoon, or ribbon, other representation methods. And also you can change the transparency of your surface, software, cartoon, or your sticks representation. For example, I would like to show the surface as a transparency with 60%. And when you click this one, and they make your surface as a transparent, and you can now see your cartoon representation of protein inside of your surface. And also you can change your cartoon with the different color to distinguish your surface and your cartoon. Okay. Let me continue with the residue by residue, maybe. Okay, let me select this residue again. And when you click this one, this cell entry, if you apply any function, if you apply any kind of action in the cell, it's directly change the appearance of this. It's directly apply this the sequence in here. Let's change this residue representation. For example, if you show this residue as the licorice, as you can see, it's change whole structure like this. But in here, it's also changed. It also showed its backbone of this residue. Let's change its color by elements. As you can see here, it's also showed its backbone. But when you open this as a licorice, it's, as you can see, there's some dot or some this kind of structure. But I don't want to see this kind of things. First of all, let's make this cartoon again, and I would like to show this main chain or the side chain options in here. If you select this side chain and click the stick, you can see it's just show this side chain of this residue, and also it gives the very nice representation of your residue like this. And as you can see, this surface is not it makes it harder to see your structure well. Because of that, I would like to hide this surface. How can I do that? In here, in the hide options, I would like to hide the surface, and now I can clearly see this residue now. And I would like to focus this residue. How can I do that? First, click this residue. In the selection, in the cell entry, there's an when it came to action, let's make it orient in here. And now you can focus this residue. Okay. And before that, I would like to show you the other things. And also I would like to show the density of this protein. And let's come here and open this 2FOFC density file. This is the X-ray density, electron density of this structure. And when you open this one with the default settings, you don't need to select anything, and you will see that this box in here, like this one, and here in the action, which is different from these structures, When you came here, you can show this density with mesh or surface or also with volume. First, I would like to show this mesh. When you click this mesh, you will see the density file, but it's kind of the looks like a nasty. But if you came here, like here, I select this one, and let's orient this one. And when I came here, as you can see this density of this residue, electron density, and also you can see other density around this residue as well. And I would like to see this this residue around to this protein, this residue. How can I do that? When you click this residue, if you click to right, if you open the, if you click, if you right, if you apply, if you right click in your mouse, you will see that there's this panel in here, and in the action section, you will see this around option. And in around option, you can select the atoms within this 4 angstrom, or you can select the residue within within the 4 angstrom around to this the residue. Let's click this residue, and now as you can see, we now select the other residues in here. And similarly, I would like to show this image, this residue as a licorice, and we should apply this cell. We should apply our action in the cell entry, not this 3-fat. If you apply anything in this 3-fat, it will apply this representation to whole structure. Let's make this structure with cell, and I would like to show this structure, this residue as a side chain. If you click this one, and as you can see, these color not nice. Maybe we can show them as a different color. For example, let's show this color as a this color. Now you can see other residue around this residue. And for example, in here we have leucine, and there's a histidine, and also in here there's a aspartic acid in here, and also there's a isoleucine in here. And we can see all this electron density of these residues as well. And in the cryo structure or cryo density map, if you see this mesh structure with similar procedure, and if you open this electron density map, and you will see the similar action in here. And if you select the mesh, and you will see this kind of things. By the way, you need to be careful, because the resolution of this structure maybe not give this kind of the nice representation. Sometimes they give the very large, or maybe you cannot distinguish well your residue. But in this resolution, we can see clearly this tyrosine in here, maybe. But in here, there's another density in here, and not we couldn't see the in the licorice as a licorice. Again, let's select around these residues. And now it's selected, but we couldn't see. As you can see here, in the sequence part, there are two oxygen atoms, and there are two oxygen atoms. First one is this one, and the second one is this one. Let me show this two oxygen with different representation. For example, let's show this oxygen as a sphere, And as now you can see this oxygen atom as a sphere. But I would like to show this one as a licorice. Now we can see that this oxygen here. And I don't want to see this density as a mesh. I would like to show the other representation of this density. For example, when you came here as a volume, when you came this action, and there's a volume option here, and when you show this density map as a volume, you can see this kind of representation here. And now it's hard to see this density actually, and I would like to change my background in here. And you can write its script in here, but I would like to show you in the displacement section. And when you came here, you can change your background in this background option here. And when you click this white, and now you can see your density much more easily as a volume. Okay. In the PyMod, you can also apply some basic calculation. For example, I would like to look the distance between this tyrosine oxygen and also this water oxygen. And in here, there's a wizard here. There's a measurement option. When you came here, you will see this panel in here, and you can measure, or you can see you can calculate the distance to ring, and also angles and the headrails of the selections. Let's continue with the distance. When you click distance, and when you see this panel in here, you need to, and also there's information here. Please click on the first atom, and when you click this one and this one, you will see this distance in here. But this label is not actually good as this label is not higher font, and you can change this font in the settings in the label here, and in the size you can change its size as you want. And here I mentioned this editing mode in the PyMod, and if you can change, if you switch your editing, and when you click this the label, and also with the control button in your keyboard, you can rotate, or you can change the position of your label like this one. And after that, be careful, you need to close your editing mode to avoid the other mislead or other unwanted action, for example. Anyway. And also, I would like to label these residues around to this residue, and let's make again select this around, and in this cell entry, let's label your residues. And now you can see this is the isolation which ID is 100, and for example here there's a isolation 78, and again you can change this label, and while you are pressing your control and using your left click. Okay. Also, I would like to show this distance to ring. You can see maybe there could be a pi stack interaction with this histidine and between this histidine and our tyrosine. And here came here the measurement, and also I'm selected this distance to ring, And first I selected histidine, and then I selected tyrosine. And now I see the distance between these two rings. Okay. What else I could say? In the visualization part, we can actually make too many representation, but the key point is here is your imagination actually, and how if you would like to if you look at represent your image with different things, actually it's available for in the PyMol. And what else I could show? And in the let's continue with the maybe. I would like to show the OPM structure in the PyMol. And when you came here to OPM, and I said download this OPM in here. And in the PyMol, if you open this OPM, you will see something like that. And here you can see your membrane position in your structure. And these are the dummy atoms, and they give this membrane location with these dummy atoms in here. And also they give these dummy atoms with different entry, and you can close it, and also you can open it again. And this is all I would like to talk about the PyMol actually. And at the end, we have this kind of, at the end, I'll show you this nice representation actually. No. Okay. Okay, this is for the PyMol section actually. I would like to show. I hope these things helps you to our further workshop section, and also your project, and also your daily life actually. And let's continue with the VMD. I'm not going to give much more detail like in the PyMol for the VMD. And let me open the VMD. When you open the VMD, you will see this interface, and also this panel in here, this displacement, this window, and here. PyMol actually, in my opinion, PyMol actually is much more suitable for the beginners, and VMD needs kind of experience, I guess. But you can start with the VMD. These are the different softwares you can use. In the VMD, like in the PyMol, there's other selection options in here, and also in the VMD you can see these extensions, and you can apply different analysis or different algorithms or different method in the VMD. You can use in the VMD. And let me first open this molecule. When you click this file and in the new molecule, you will see this browser in here. And let's click here. And let's open our PDB. And in the VMD, they show you which data format they can provide, they can support. and you can select by manually in here, or you can select automatically in here. When you load this structure, you will see this line format, line representation of your structure. And first thing you need to do is change your representation. By the way, I need to load this 3FAT. Not the this one. And when you open the tool file like this one, you will see the tool file in your workspace. And by the way, in the PyMol, you can change your rotation, or you can transform your structure with nice with your mouse, like clicking right click and left click. But in the VMD, you need to use this mouse section here to translate your selection or your rotate. For example, when you click this translate, you can translate your structure, and you need to change your mode as a rotated again to rotate your structure. And in here, there's a selection modes in here that's top, and this is the action when you apply this one, for example. Okay. Anyway, in the D represent the displacement. And here, if you click this one, you hide your this entry in here. And in here, in the graphics, you open the graphical representation section, and you will see this is actually most used part of the VMD in the visualization. You can create representation and also change the appearance of the structure. And here there's a drawing method. And if you select the drawing method as a new cartoon, you will see this kind of representation in here. And you can change your coloring with this, for example, with this coloring actually. And in the selecting atom part, you can select select your residue like this one. And if you write this all, it represents all. And for example, let's delete this one and show select the res ID in here. And for example, let's come here again the 88. And let's apply. And as you can see, we see this cartoon, but let's change its appearance as a licorice. And now you can see this tyrosine. I guess, what time is it? I guess I have 15 minutes. Okay. Let me show more about the VMD. And let's create another representation. And let's write a protein. And now we see the all protein in like this one. And also we can change this coloring with the name. And it changed the coloring with your atoms. And let's change this representation as a cartoon. And now what you see, your first representation and also the second representation in the same image. And in here, when you select the res ID, you will see its backbone as well. And let's hide its backbone. And here you need to select. You need to say and I don't want to add this backbone. And you define the single word backbone. And now you hide your backbone, but you hide also your alpha carbon, and you need to say in here maybe the name alpha carbon. Let's say name JA, name CA, represent the alpha carbon CA. And now what we see, stop. Okay, my computer is. Okay, now what we do, we open the residue 88 and we hide the backbone, and then we show the alpha carbon again. And now, as you can see here, we selected this residue, and let's make some measurement in the VMD. And it's very maybe difficult when compared to PyMol. But when you came here, the mouse, and let's select the label here, and you can select the atoms, bond, angle, dihedral. By using this selection, you can measure, or you can measure the distance between the atoms, or you can measure the angles or dihedral. Let's select this atoms, and I would like to select, for example, this one, and you can see this automatically labeled this tyrosine oxygen in here. And okay, maybe we can measure distance between this alanine or the other residue. And in here, I would like to show the other selection in here. If you select one residue, you need to write this 88. But if you would like to select more than one residue, you need to write the 2, and you need to define the other. You need to define the range like this one, 88 to 120, and it will show you all the residue within this range. For example, and also you can define your residue like this one, like given your residue ID using the white space. For example, 95, 90. What was the name of the histidine? 99, 97, I guess. 96. Okay, 96. Okay, I would like to delete first this label. And how can you do that? If you came here in the graphics in here, there's a labels option. When you open this label option, you will see the all selected label in here. You need to delete if you would like to delete this label, or you can show or hide this label. And I came here again in this label section, and I'm selected the bonds. And let's make this kind of look. And select the label and the bonds. And I'm selecting my first atom, and I'm selecting my second atom. And now it's automatically show your distance between these two atoms in here. And also you can see this information in your labels window in here when you came here and select this bonds. And now it's gave the information about this distance measurement. It gave this value, which is your distance between this atom. And what else we can do in the VMD.
Hi, Cemilcan.
Okay.
I would like to remind you that you have about 10 minutes left and we have some questions in the chat box.
Okay, I'm finished here and I'm continuing with the this chat, the questions.
All right.
Oh, wait a minute. How can we undo action in the PyMol? Actually, there's a command like undo. If you type in the command line, let me show you. But sometimes it's not working well, but this is the way to make undo. And there's a command like a undo, and it should be gives you the previous action actually. But sometimes it's not working well. I actually when I did something wrong, I'm start over. I'm start from the beginning actually. How can we show the names of the interacting residues of docked complex in the PyMol? I showed you this around and the label in here. You can select your ligand, and you can click your ligand, and you can select the around your ligand, and then it will be create a new entry, and you can label this your structure. Could you please show again the density map? Let me show you. And in the here, I opened this tree fat, and I downloaded this density map in the Protein Databank. Protein Databank, yes. Tree fat. Let me open this one. In the download files, I said I download this 2FOFC map in here. And these are also the density map, but the calculations are different. And when you came here, let me open here. And this is the downloaded file. And if you open this one, you will see this kind of map import section. You can directly load your density with default options. And you will see this the box in here. And there is an action here. You can represent as a mesh or the volume. If you select the volume, you will see this densities. And if you focus one of your residues, for example, this one, if you show this alanine as a licorice, for example, you will see this kind of things. How can you show the interaction of protein with the silver nanoparticles? You can use the PyMol scripting actually. There are many different scripting, and they also show the interaction interface of the proteins and other selection of your proteins or the nanoparticles. You can use the PyMol scripting and Unity. Actually, you can browse and you can try the PyMol scripting. I didn't show the PyMol scripting because it could be more advanced actually. What are the key differences between PyMol and VMD? It's hard to say actually because these are the different programs and they provide different properties actually. In the VMD, for example, you can, it's VMD, for example, direct, it's much more easy to combine with the EMD molecular dynamic simulation, for example. In the PyMol, you can actually see this kind of things. And in the VMD, for example, you cannot open this density file like by clicking this kind of things. For example, in the VMD, let's try the file. It's not going to give, for example, let's open this one. Oh, it's open, but it's not open this compressed file, but it's also open this density map in here. But my VMD is stuck. They give the different properties actually. How we add the new bond in PyMol and VMD. If you can use the, I didn't know in the VMD, but in the, let me close this density map in the, okay. You can use this density, you can use the editing mode in the PyMol. For example, let's try one example maybe. Now you can see your structure. Let's click this editing mode, and you should select these two structure, these two atom, and there's a build option here, and you need to create bond in here. If you click this one, it's create the bond between these two atoms. And after that, you need to close to this editing, because, for example, let me show you. Close this too. Let me show you how you can use the editing mode. Let me come here. For example, my editing mode is open now, and when I click this one, and I'm pressing my control button, and I can rotate, or I can move my this rotation, this dihedral in here, and to change my structure. And if you make this one, if you do this kind of things extensively, you actually make your structure worst maybe. Since both are visualized molecules, yes, these there are different opportunities, different properties actually. You need to read, and you need to use what makes you comfortable actually. But in the structural biology, we all use these softwares, and sometimes I'm use the PyMol, sometimes I'm use VMD, sometimes I'm use different software like Chimera. How to add the ions and its coordinate. You can use in the PyMol, you can use the builds here, and you can add the ethyl ion maybe like this. For example, let me close this one. You can add the fragment like acetyl or the methane, for example, like this. And you can create new fragments, or we can add different things. But if you ask, yes, you can visualize the RNA protein complex in the PyMol or VMD. It's support. If you are asking the adding ions for the molecular dynamic simulation, you should use the VMD, and there's a plugin and there's a method to add ions. You can use this one. Okay, I guess that's enough. And there are no questions. You are welcome. I'm stopping my screen. I stop my screen share now. Thank you for listening.
Thank you very much for your very nice explanations and presentation. Our next session will begin at 1 hour later, 1 p.m. GMT +3. And before going to a lunch break, I would like to remind you that after the lunch break, we will have a presentation titled Molecular and Micromolecular Docking by Assistant Professor Salia Ece Acuner. And yeah, now it's time for lunch break. See you in 1 hour.